our clinical 10 subjects. So we wanted to look at the what are the anatomical features that influence the TMS response, that is RMT, as well as what are the functional features. So we not only took their MRIs, but also we took DTR, that is diffusion tensor imaging. So basically, it's a specialized protocol in MRI equipment. When you get this MRI image, you can use this protocol to measure the water diffusion in the brain, and water diffuses along the neuron. So you get these nice fiber tracts based on the water diffusion, and these fiber tracts tell you which brain areas are connected to other areas. So you can consider this as a structural connectivity of the brain. So this could be different for different people. So again, the structural connectivity depends on how your brain is formed and how there has been brain plasticity, and all those parameters will change your DTI image. Yeah.
Is there a reason for different colors? Is it like different color agents?
So yeah, we just there is no meaning for different colors. We just showed different colors just to distinguish. Yeah, and you can see the shapes of each person's brain is very different. So the brain scar distance is different from person to person. So when you take the dose from motor cortex and use the same dose to stimulate the executive part, emotional part of the brain to treat depression, you might not be doing it right, because the brain scar distance at the motor cortex is different than the brain scar distance at the executive part of the brain. So that means the dose that you measure on the motor cortex may not be the right dose for firing the neurons in executive part of the brain. So, but in the clinical setting, people don't look at MRI, they just use this protocol, they get the dose here, and you apply it on the executive part of the brain. But it still works. So now, if you have MRIs, you can refine it further and make it more effective. And also, that's just the anatomical features, that's just the geometry. But also the function, the structural connectivity can also be used to determine how the structural connectivity will influence the dose parameter. So that's what we looked at, and we used linear mixed effect modeling to see how different parameters affect the RMT, which is the dose. So now again, we built the head models, and we took their RMT, whatever RMT they had, whatever dose they had, and for that dose we determined their induced electric field, and we compared the induced electric field to their brain scalp distance. You can see there is a nice correlation. I can't see the correlation coefficient here, it's quite high. Yeah, this is as expected. We have seen it in public database as well. So the brain scalp distance changes your dose, which is taken care when they determine the dose, when they apply it. That is taken care when they determine the dose. But now, if you look at their functional connectivity, that is, if you look at their fiber tract density, the fiber's density, that will also influence the dose. So here, what we see is for low surface area or low fiber density, we looked at the surface area as well as fiber density. For high surface area, it is as expected. as the RMT decreases, the volume of gray matter, the volume of brain depolarizing or volume of brain being stimulated above the threshold decreases. But for lower surface area of the fiber tracts or the fiber density, the trend reverses. And for medium, it's not so, and also we had only 10 subjects, so it may not be indicative. There might not be a statistical significance here, but within these 10 subjects, we see wide variation. What that tells us is, yes, the structure of the brain influences your dose, which is taken care in clinic, but there are other parameters which are functional or structural connectivity, and also other functional connectivity parameters, which is, I'll talk about it later. But the structural connectivity does influence your dose parameter. So, but if they had to take the DTI image, it's very expensive, it's time consuming, but yeah, which could, if you take DTI and consider that, you might increase the effectiveness of the treatment. So that's about healthy subjects. And in 5 minutes, I have to complete 2 other clinical trials. So this is traumatic brain injury, and these are the veterans who had a brain injury from our Richmond Veteran Affairs Hospital. So there were, again, this is in collaboration with Catherine Holloway, the neurosurgeon. So she contacted about 1,434 veterans who had TBI, and out of them only 550 did not agree, and only 350 agreed. Out of those 350, only few, that is 26 people, had the criteria, which means because we wanted only TBI, they had other comorbidities, it would make the study complex. So only 26 were selected, and the original study was to look at if we can see any variation in cognitive scores pre and post TMS. That was the original study, but I was more interested in coil design and how the response, that is RMT, is influenced by other factors. So we looked at their brain scalp distance, and again matches the trend. We also looked here, it is E field, maximum E field and brain scalp distance. Here this is RMT and brain scalp distance. As the brain scalp distance increases, the dose increases, the intensity increases. So that is as expected. But we also took their EEG pre and post. So in one study, which I did not put here, we show that post TMS there is high delta power. So when you look at all the EEG signals, you can calculate the power for each wave. There is beta, alpha, delta. And when we look at specifically the delta waves, the delta power increased. What exactly that means, we don't know, but there was increase in delta power post TMS treatment. But I was more keen on how the functional connectivities between different regions of the brain influence the TMS response, in addition to the geometry, anatomical features. So that's what we looked at. Again, we used mixed linear effect models, and we show that here. So that's not very clearly visible. This is the DFP1 connectivity. So basically the connectivity of the emotional part with the motor cortex. So this is C3, C4 connectivity. This is C3 and C4, or the bilateral, so the connection between two hemispheres. and then PF1 FOOF measurement. This is another new parameter that people are excited. This is our Harvard collaborator who wanted to look at this parameter. Apparently that tells about how much you can inhibit the prefrontal cortex. So this is another parameter that is of importance. We looked at all these functional connectivity. So the dose not only depends on the geometry, but also the functional state. And how do you measure the functional state? You measure by structural connectivity or the functional connectivity. Functional connectivity can be determined by EEG or fMRI. And we looked at EEG functional connectivity in the TBI subjects, and show here that functional connectivity is also an important parameter that needs to be considered while determining the dose. And then we looked at, this is a clinical trial at Nimans, India. So here they had close to 100 subjects, 40 healthy and 60 schizophrenia subjects. And we show that with the healthy population, the correlation between the geometry and the dose is better than the correlation between the same parameters in schizophrenia subjects. What that means is in schizophrenia patients, the functional connectivity is altered more. So that's why when you're treating with patients with certain disorder, not only the functional parameters are more important as well. So, and here we look at the functional connectivity of these schizophrenia subjects. This is an fMRI study. Basically, this is fMRI is looking at oxygen perfusion in different areas of the brain. So these are all different areas, and of course, this is how they are normally plotted. The red, for example, this of course has to be red. This is correlated to the same region. But if we look at this one, this is IC22 to IC3 has a good correlation. That means when you stimulate IC3 region, you end up making IC22 region more active. So this is the functional connectivity map, and we looked at how different regions are connected and how these parameters influence the dose of TMS. And again, different connectivities will influence differently, so we need to consider all these parameters. There are so many parameters, and all these parameters need to be considered in order to determine the dose. And what's the best technique to do that? So of course, machine learning. So we are using machine learning to predict. So we give all these parameters, and we have so much data. We train our machine learning models and predict first induced electric field, just using the anatomy, and then we use functional states also as one of the input parameter for predicting the dose. And that is still work in progress. I think it's 10:30 now. I will conclude. I'll be around for taking any questions. Here are my conclusions, and I would like to thank all my funding agency and my postdocs and students who have worked on these different projects. Thank you.
Thank you for the nice talk. I have a question like, are there any known side effects? Like you mentioned the old method, there are like memory loss. Is like something like that also present in this method?
For TM, there are no no side effects or adverse events reported apart from seizures, short episodes of seizures in patients who had history of seizures.
Okay, thank you. Maybe one second question. You mentioned also that there are eye neurons. What happens if you trigger them, so that you excite them? Have you like hallucinations, or do you don't see any difference?
Well, it depends on which area you target.
Yeah.
So if you target motor area, it just
Yeah, for example, this would be the neurons that are proposed to have like the eyesight.
oh, eyesight.
Yeah.
So you get phosphine effects. So you get phosphine, so that you see bright light. And that is actually one of the people have done a lot of phosphine studies.
Okay, thank you.
So, Professor, I had two questions. So the first is, has the correlation between age and brain to skull distance been established? Like, can TMS be used for growing children?
Well, for that you don't need TMS, right? You just need MRIs. And if you look at MRIs, you look at different age and you can measure all the parameters.
So then TMS is like adaptable for growing children, or is there a different protocol? Or?
No, it's not FDA approved for
Growing children?
Children or teenagers. So there is a lot of need, a lot of inquiries from many people.
Because there is like childhood depression. And,
Yeah, so, and there is, a lot of teenage issues also. So, that's an active study. A lot of people are looking into it. Again, this because the developing brain has varying, not just anatomical parameters, but also functional connectivities. It's not fully developed. Even if you take the functional connectivity, it might be something today, and in 1 or 2 years it might be different. So it's an ongoing study.
Okay, and the second question is it adaptable for people with congenital brain abnormalities? Like, for example, some people might be, so I've heard, some people might miss the corpus callosum that connects the two hemispheres. So in such cases, is it, again, contraindicated?
Okay. So again, so how the different anatomical variations influence the E field and dose need to be determined. And then it's not just anatomical variation or anatomical features, but also functional connectivity. If their anatomical variation changes their functional connectivity, and if they're still normal, so those parameters need to be considered.
So it's adaptable basically for healthy adults who are facing
It's easy
functional.
so it's FDA approved for adults, yes.
Thank you.
Are there studies combining magnetic nanoparticles to enhance local TMS?
Yeah, so that's one of our interests too. So there is what is the magnetoelectric brain stimulation. I think from University of Miami, there is a student from University of Miami. So they are working on magnetoelectric effect. So you can put nanoparticles in specific regions and use the magnetoelectric coupling in these nanoparticles. So the core is a magnetostrictive, and the shell is piezoelectric or ferroelectric material. So when you apply fields, the core changes the dimension, that changes the potential of the shell, and that induces the voltage in the neurons. That's called as magnetoelectric effect. But for that, you need to inject nanoparticles where you need, or you need to have nanoparticles where you need to stimulate. And that is another area of study.
I have a curiosity about the phantom that you made with PDMS and carbon nanotubes to make it conductive. And I was wondering if there is a particular reason why you decided to use PDMS with carbon nanotubes instead of a polymer that is already conductive by itself.
So the human brain is quite large. So silver nanowires actually work better, but they're very expensive. And if you want to put it in a large volume, so we just chose the most cost-effective material.
Thank you.
Professor, so I would like to know about the cost of this treatment as compared to other treatments for Parkinson's disease, and how it is going to be in the developing nations. So they are means.
So yeah, if you think about the device and the coil, it's just a capacitor and then a CR or a thyristor to deliver the large current in a copper winding. As such, the device should not be very expensive, but because of proprietary rights, they are. You can the cheapest is $35,000 US. Earlier they were selling at $130,000 US, but you can build a much cheaper one as well. So it should not. If there is a will, there are teams that are working. It's not difficult to bring it to masses.
Thank you, Prof.
Thank you. This was very informative, Ravi. How do you know how much magnetic field to apply? How do you know which one, 2 Tesla, 3 Tesla, 5 Tesla? Why stop there if it's useful?
Yeah, so we want to basically depolarize neuron membrane to make them fire. So what field makes them, so at 2 point, okay, I think I know it. So dB by dt. So you need a high frequency. If you go at very high frequency, you get high field for a low magnetic field, right? But neurons don't respond beyond 5 kilohertz because that's the time constant of the ion exchange between the membrane. So you have to stay below 3 kilohertz, and at 3 kilohertz, if you want to get 150 volts per meter, you need about 2 Tesla on the surface of the coil.
Thank you.
You're welcome.
Any more questions?
So I'll be around if you have more questions. I can answer.